Hi friends, welcome to HMP Shunt Pathway Part 1. In this part, we will be discussing the significance of Hexose Monophosphate Shunt Pathway. So HMP or Hexose Monophosphate Shunt Pathway, because it involves hexoses like glucose 6-phosphate and fructose 6-phosphate, and shunt pathway because this process involves recycling of metabolic substrates. Pentose phosphate pathway, it involves pentoses like ribose, ribulose and cellulose. Has also 6-phosphogluconate pathway because this involves obviously this particular substrate and it gets oxidized also. Now, this, as you know, it's the first part of glycolysis or the preparatory phase of glycolysis. Starts with the glucose and it goes through glyceraldehyde 3-phosphate. We know that this will continue and give pyruvic acid finally. But here, there are three substrates: glucose 6-phosphate, fructose 1,6-bisphosphate, and glyceraldehyde 3-phosphate. Normally, in a cell, nearly 90%, 70 to 90% of the glucose will be channeled to glycolysis for the production of ATP. But there is an alternative pathway through which nearly 10 to 30%, depending on the cell, 10 to 30% of the glucose passes through or goes through this alternative pathway. That is what we are discussing, pentose phosphate pathway. Now, normally 10% of the glucose enter the pentose phosphate pathway, whereas in some cells like tissues like liver or RBC, which use the substrates or the products of pentose phosphate pathway in a greater amount, they will consume nearly 30% of the total glucose available to channel to the pentose phosphate pathway. Now, there is fructose 6-phosphate and glyceraldehyde 3-phosphate, which can be taken to pentose phosphate pathway from glycolysis, as well as they can be taken from PPP to glycolysis also. So glycolysis and pentose phosphate pathway, the substrates involved are interchanged, or they can be mutually taken and given. Now, actually, in a cell, the cell do not know that whether the substrates present in the cell cytoplasm or the cell pool, whether they belong to PPP or glycolysis, as we have termed them. What the cell does is that at that particular moment of the time, which one is more necessary, whether glycolysis and ATP is necessary, or pentose phosphate pathway and NADPH is necessary, or production of ribose is necessary. So it depends on the need of the cell that these substrates will be taken to whether to glycolysis or to pentose phosphate pathway. Before we discuss the significance of HMP, we can go through what actually HMP means. This is divided into two phases: oxidative irreversible phase and non-oxidative reversible phase. So none of these reactions are reversible. Here in this first phase, these reactions are but reversible. Here in oxidative phase, we can see the NADPH, 1 molecule and 2 molecule. So this is the most important product of oxidative phase because this is called reductive potential, which is involved in reductive biosynthesis of a number of substrates. So that we will discuss later. And non-oxidative phase involves the production of ribose 5-phosphate. Yes, ribose 5-phosphate. Now, ribose is involved in the production or synthesis of nucleotides. So this is ribose is precursor of nucleotides, which means they are involved in the synthesis of DNA, RNA, and high energy molecules like ATP, GTP, then NADH, all these molecules. So this is, and non-oxidative phase is reversible. So all these steps can be reversed to give rise to ribose 5-phosphate also. Now the first part, oxidative phase. So this is concerned with production of NADPH, the reducing potential. Now, NADPH is involved in reductive biosynthesis of cholesterol, fatty acid, and so all the molecules which are derived from cholesterol and fatty acid also, like steroid or let us say phospholipid or triglyceride. All these molecules depend on NADPH for their production, basically because they are involved. NADPH is used up during cholesterol biosynthesis and fatty acid biosynthesis. And so these tissues which are specialized in the production of these substances, or the function of which are listed here, will be using NADPH, or will have a higher rate of this particular process, that is HMP pathway in them. Like you can see here, this steroid synthesis or fatty acid synthesis or steroid synthesis which happens in ovary or testis, all these things. Now here one more, red blood cells. NADPH is involved in the maintenance of reduced glutathione. So this will be discussed in the slides to follow. The second part is non-oxidative phase, which is present or which is taking place mostly in all tissues, because all tissues require the special product ribose, which is the part of non-oxidative phase of HMP pathway. Thus, especially this is in high demand in cells which are actively growing. Synthesis of ribose is possible by reversal of pentose phosphate pathway also, as I said. The non-oxidative pathway is reversible. So any of the molecules, any of the substrates involved can be reversed to give rise to ribose. As also there are a number of substrates involved which are having C3, C4, C6 or C7 carbon numbers, like one of them is glycerol A3 phosphate, and the other one which is having 7 carbons in it is pseudoheptulose 7 phosphate. So all these molecules are involved, and rapidly dividing cells of course require more ribose 5 phosphate than NADPH for obvious reasons. Yeah, now we can get more familiarized with the structure of NADPH. The structure of NAD plus is shown here. Nicotinamide adenine dinucleotide. We can examine whether all these molecules are there in this big structure. First of all, adenine, yeah, of course it's there. Ribose is there, an additional ribose here also, because adenine dinucleotide. Nicotinamide adenine dinucleotide, and they are connected through pyrophosphate, and this is nicotinamide. This is the most reactive part of NAD plus, most reactive part, and nicotinamide ring is the reactive part. This is because in the oxidized form, the nitrogen atom here carries a positive charge. That's why this is indicated as NAD plus also, the positive sign. And here when NAD plus has a phosphate group esterified to the second position of one of the ribose here, and the second position esterified with the phosphate group, then it becomes NADP plus. Now, even though NAD and NADP are having similar structures, they are not interconvertible. And they are not formed through same chain of reactions also. Not formed through same synthetic reactions. Now, during oxidation of substrate, nicotinamide ring of NADP plus accepts a hydrogen ion and 2 electrons here. It accepts a hydrogen ion and 2 electrons, which are equivalent to a hydride ion, and now NADP plus is reduced to NADPH. So this is what happened, and the valency of nitrogen is reinstated. The valency of nitrogen is 3 is reinstated, as well as the electrons are accepted here, as I said. Now, the high potential electrons are required for most biosynthetic reactions, because most of the biosynthetic reactions, substrates are more oxidized than products. So that is there is the role of NADPH as the donor of electrons during biosynthesis, since substrates are more oxidized than products. And NAD plus also, NAD plus accepts a hydrogen just as NADP plus accepted. NAD plus also accepts hydrogen ion and 2 electrons to become NADH, just like NADP plus has accepted 2 electrons to become NADPH. But NADH has an entirely different role in metabolism, and NADH are actually used for synthesis of ATP through oxidative phosphorylation. So this slide shows the structure of NAD plus, NADH and NADPH. You can pause and study it well. Now the significance or the role of NADPH in cellular metabolism. The first one we have already discussed, the reductive biosynthesis of fatty acids, cholesterol and steroids, and the second one also mentioned in one of the earlier slides, that is NADPH maintaining the erythrocyte cellular integrity or membrane integrity. In fact, NADPH is required to keep glutathione in the reduced state, and which is responsible, that is reduced glutathione is responsible for the detoxification of free radicals. So, now what is glutathione and what are free radicals? Now, glutathione exists in reduced GSH and oxidized GSSG states or forms. The ratio of reduced glutathione, this one, to oxidized glutathione within cells is a measure of the cellular oxidative stress. Whereas increased GSSG to GSH ratio is indicative of greater oxidative stress, which means if GSSG is high, that means high is the or greater is the oxidative stress. In healthy cells and tissues, more than 90% of the total glutathione pool is in the reduced form. 90% is in the reduced form, with the remainder in the disulfide form. This is the remainder. The oxidized state is converted to reduced state by NADPH and the help of NADPH, and this conversion is catalyzed by glutathione reductase enzyme, as shown here. The antioxidant GSH actually protects cells by neutralizing reactive oxygen species, neutralizing reactive oxygen or reducing reactive oxygen species. This conversion is illustrated here by reduction of hydrogen peroxide to water, and this slide illustrates further. You can see GSH here, GSSG, and GSH is actually responsible for scavenging the free radicals. Here hydroxyl free radical is shown here, thereby protecting oxidative damage to biomolecules as well as to cell membrane, and in the process GSH actually gets converted to GSSG in the presence of glutathione peroxidase, and GSH level is reinstated with the help of the enzyme glutathione reductase, and here NADPH is the electron donor, and NADP is formed or NADP plus is formed, and this level NADPH level is reinstated with the help of this particular metabolic process which we discuss, HMP pathway. So this is an exclusive slide. You can pause and learn it well, how superoxide radicals are formed, how hydrogen peroxide is formed, and hydroxyl free radicals are scavenged by GSH, all been illustrated in this slide. And continuing with the significance of NADPH, the prevention, this involves the prevention of methemoglobinemia, and this is NADPH is required to keep ferrous ion of hemoglobin in the reduced ferrous ion stage. Then it keeps the transparency of lens of eye also. Actually, NADPH is needed for maintaining transparency of cells, and this is being done through the glutathione involved process. When NADPH has got a role of activating the antioxidant GSH, at the same time it has a role of producing reactive oxygen species molecules also. But this time it is to protect cells in another way. So as shown in this figure, NADPH oxidase catalyzes the production of superoxide radicals. You can see the superoxide radical indicated here, oxygen with a negative sign and a dot there to indicate extra electron. So this free radicals, this is by transferring one electron. This free radicals are formed by transferring one electron to oxygen from NADPH. During this process, oxygen is actually transported from extracellular space to the interior of the cell so as to produce superoxide molecules. Now, superoxide thus formed in phagosomes. So this is actually formed in phagosomes and macrophages. So it can counter bacteria and fungi. Superoxide spontaneously gets converted to peroxide, which is then protonated to produce hydrogen peroxide. It is thought that the hydrogen peroxide and then hypochlorous acid formed are actually responsible for lysing bacterial cells. Now another role is in photosynthetic organisms. The green plants, where NADPH is produced in light reaction and utilized in dark reaction so as to reduce carbon dioxide step by step to produce sugar molecules. So that's what happens in photosynthesis.